Hey guys, good morning. Good morning, families. Good morning, people. Thank you for being here. We are going to, Pavel and I'm JR. Pavel and I are going to talk a little bit, just a very little bit about how amazing Science Day was yesterday. And then we are going to answer your questions. So I want to say to the room, um, if you have a question, like come up close and talk to us. I think there's one roving mic maybe, and it's with Ed back there. And if you are at home on the Zoom, where's the camera? If you're at home on the Zoom, yeah, put your questions in the chat. We're going to try to answer them. I have this whiteboard up here, and it turns out that writing on a whiteboard is a terrible idea. So I'll probably have to talk, but please ask your questions.
Hi, I guess it's a quick intro. I'm not as famous as JR, so people might not know me. Yeah, so I'm Pavel. I work at SRF and do partnerships and grants and things like that. So I do a lot of the grants to the institutions and work with some of the pharma companies as well. I'm not a scientist. I'm just a person who tries to read papers on occasion. So I'll definitely be deferring to JR on the sciencey stuff.
Pavel is being way too humble. He understands every grant. And when I don't know which grant is which, I'm like, Pavel, what's happening? He tells me. So he's being way too humble. But I'm the one who asks the really annoying questions. I'm the one who asks the questions that really get into the weeds. So we're a good team because he understands the whole structure of what's happening. And I'm the one who's like, but wait, what about? Okay. So we have one overarching message about yesterday, and that is moving fast. Like we are really, really moving fast. This organization has done amazing things. Thank you to everybody who's part of SRF, everybody who's ever given to SRF, everybody who's partnered with or worked with SRF, anyone who's answered emails and reached out to us. We are really, really moving fast. And Mike said it really well yesterday. So you're going to have to look at the webinar when it's out on webinar of his closing comments. But his closing comments are just spot on. Like we, in every arena, we are moving fast. And so I think there's what, Pavel, there's here, you're the structure guy. There's basic science, trying to understand what's happening. Um, we're all related to each other by a gene SYNGAP1. And then we're kind of tangentially related to a whole bunch of other people who have a neurodevelopmental disorder, right? And in the neurodevelopmental disorder family, let's say monogenic neurodevelopmental disorder family, so that's a one gene, on your genetic report, you've got one gene problem. Within that, we're kind of related to the people that have a synaptopathy, so where the synapse is the issue, right? So our synapse is the issue with SYNGAP1. The SYN in SYNGAP stands for synapse, which is SYNAPSE. Yeah, you're spell checking. I got spell check right here. Okay, so we have these sort of like circles of relatedness to each other. And our relatedness is through the SYNGAP1 gene, right? So we kind of have to understand what's going on. That's the basic science side. Then what are the other sides, Pavel?
Yeah, so you have the basic science piece and then obviously like how do you translate that basic science into treatments? So the really deep things that Marcelo Koba and Marcelo Bertelli presented yesterday, the next step there is like how do we use this knowledge to create things that will modify the disease and increase the levels of SYNGAP or fix some of the things that are broken because there's not enough SYNGAP. So that's sort of the translational piece, and then the where the rubber hits the road is getting those treatments into the kids. And yeah, I think the day yesterday was fantastic. We're doing really really well. I mean, for me, it's been a journey because when I started, I was like, "Oh, there's this thing that can cure SYNGAP. Like the technology exists. We're done." I was so excited, and then it turns out, okay, well, we have a bunch of clinical data, and seeing what we saw yesterday with the clinical data we really did not have much. I mean, we had almost nothing when we started a few days ago. And now we have tons and tons of data. Dr. Levin's presentation with the EEGs at the end, I thought was really, really impressive. Like, they're using the EEGs and they're getting data that's unique to SYNGAP kids. And immediately my thought is, okay, if we give these kids a treatment, this thing's going to get better. Like, we can show that to the FDA. That's so important. And then I guess what I'm starting to learn now is the next bit of this is really dealing with the regulators and dealing with the FDA, and that's the next thing we have to tackle. So I think we're moving really, really quickly. We're making really, really good progress, and the path is like laying out before us on how we can start getting these treatments into the kids.
Yeah, it's true. It's like, okay, if the science exists, why aren't we doing it, right? What are the holdups? And there's a whole bunch of them, and we heard about a bunch of them yesterday. The thing that I'm really excited about right now, and that is always top of mind for me, is the measurable outcomes. And so the issue is, what if we understand enough of the biology to have a good treatment plan, and then we have developed the treatment, and it's got a good chance of working, and we have some clinical measures, and we think they're going to work. Well, there's still a chance. There's still like a terrible chance that a trial could fail, right? When a clinical trial fails, it doesn't necessarily mean that the treatment doesn't work. It means that the trial failed. The trial design really matters. The trial design includes who is in the trial, how long are they in the trial, how long are you waiting between what you measure, what are you measuring? What if you know it's going to make a change in something, but what you measure didn't change soon enough? Well, the trial's going to fail, right? And so there's a lot of drugs that are potentially good drugs, not for SYNGAP. I'm just talking in general. In general, there are drugs that are potentially good drugs that have failed not for the reason that the drug wasn't helpful, but for the reason that the design wasn't good enough. So we really want to make sure that we understand, and I think this is partly what Pavel's talking about with the regulators. We want to understand what is the way to make a trial design, not to make sure, here's where I'm going to get really picky about wording. It's not to make sure that the trial succeeds, it's to make sure that if the treatment is good, the trial succeeds. Okay, sometimes trials fail because the drug doesn't work, right? If it's not helping, we don't want it to succeed, right? We don't want to put something in our kids that doesn't help. Okay, so there's a couple reasons why a trial could succeed or fail. We want a meaningful treatment that is successful, that has a successful trial. That's what we want. That's how we're going to get there.
Yep. So I don't think we want to like sit up here and talk. I think, we really appreciate that the families have come here with the kids. We know the travel is really tough, and for those that managed to catch some of the science day yesterday, I know there's a lot of dense content in there. So I think we really just wanted this to be more of a Q&A and really give people the opportunity to ask like any questions they have at any level, and we'll do the best we can to answer them.
Yeah, just say any level again.
Yeah, any level. Please don't be shy. Like, we'll do our best. And if it's too advanced, then we have actual researchers here. There's nothing too advanced for JR. I don't know why I'm saying that. Yeah, that's fine. So Ed's got a mic, and then we'll keep an eye out on the Zoom as well if there's any questions.
Thank you. Hi. So not particularly related to any one talk yesterday, but a question I get a lot from new parents is when they hear that it's rescuable. So when they hear that, sort of in frogs or zebrafish, or, sort of the disease has been rescued or it's reversible, I'm always very cautious with that because, so I have a 7-year-old, his brain is developing at an age, I really don't think that, we're going to give him a drug and he's going to turn into a neurotypical 7-year-old. Can you talk a little bit about what, sort of what it means to rescue, so potentially increasing SYNGAP for seizures or for other aspects? Just to sort of give a bit more color around that.
So I'll take a stab at it. So I think there's real data points that we have. So Gavin Rumbaugh had a paper where he reintroduced Syngap into older mice, and the mice improved. Their seizure profile improved significantly, so they got better. So he demonstrated that regardless of the age of the animal, there is an improvement in reintroducing the Syngap. And he talked at the conference last year and said neurons really need their Syngap, or synapses really need their Syngap. So it's going to help. That's what we know. What we don't know is obviously in humans how much it's going to help. And also what we don't know enough about is the downstream pieces. So when there's not enough Syngap, Dr. Wilsey presented about cilia yesterday, like things get messed up downstream, and Clement Chow's fly screen yesterday was a good example of this. He took away the Syngap, the fly's eye became disformed, and then he introduced some drugs, and the eye got better. That may be because there's more Syngap, but that may be because whatever was getting broken because the Syngap wasn't there got better. And so what we need to try to understand, and what we're doing with a lot of the small molecule work on some of these grants, is there a way to improve the things that got messed up because there wasn't enough Syngap? And is it going to be possible to do that in the older kids? And I mean, I've got a 6-year-old. I certainly hope that that's the case. And I haven't seen anything that shows it's not the case, that we couldn't have a significant rescue of some of these phenotypes.
Yeah, I mean, Pavel said it great. I think Gavin's paper used the Crelox. Is that right? He rescued it.
Yeah, the one that switches.
Yeah. So I think in Gavin's paper, which is it's really important to see that he was measuring specific things that are rescuable. There are other things you can measure that aren't rescuable. So if the thing already happened and is never going to happen again, you're not going to rescue that because the process isn't in play anymore, right? And so he was able to use a genetic tool kind of methodology to have mice that started out without it and then switched them so that they got it back. That is not a treatment option that's available to us. And so the way because we don't have our kids don't have that magic thing in them right now, right? They don't have that genetic trick that you can do. They don't have that tool in them. So the way we deliver drugs to our kids, whatever the treatment will be, that'll partly matter as far as what gets fixed. So if we are thinking that, I mean, Syngap clearly is a huge effect in the brain, and it obviously has a huge effect in excitatory and inhibitory cortical neurons and other cell types in the brain. But we also know that there's gut issues, and so like Julia Dahlman at University of Miami is studying zebrafish gut, and she's shown really beautifully what happens when the fish don't have one of their Syngap genes in the gut, and the peristalsis that makes the stuff move through the fish. It's all messed up, and the stuff kind of just sits there. It doesn't like travel. So if we treat the brain and don't treat the cells involved in that, I don't really see how that is going to get fixed. So part of why I'm so very, very excited about Dr. Wilsey's work is because it's not because I think that in our Syngapians all their cilia are broken. That's not true. It's not possible. They wouldn't survive that. So that's not it. It's that I think some of the features that I see in my son are more likely attributable to a cilia issue somewhere than to an excitatory or inhibitory cortical synapse issue. And so my goal would be to understand when and where is SYNGAP over all stages, over all tissues, and when and where is having half the amount okay? When is that fine for a cell or a tissue? And when is having half not okay? And so where having half is not okay, that's where we want to treat it. And it might be more than one or two places. It might be more than just the brain part. And we don't, yeah, and like I said, we don't have the magical mouse trick that you can just flip it on everywhere. I mean, yeah, if we did, we wouldn't sit here. We'd be at home doing whatever we want to do.
Yeah, so I think to answer your question in one sentence, there is evidence that there's hard evidence that patients of all ages can improve with treatment, and we need to get the treatments out there. Question
100%,
over here.
100%.
Yes, I hear you talking a lot about deletions, but what about additions, like duplications and stuff like that? Is there progress on that side as well?
So you're talking about an entire gene duplication?
Like just sections of the gene to duplicate?
Yeah. So if you have a small meaning less than the size of the gene, if you have a deletion or an insertion that's smaller than the size of the entire gene, it's gonna make basically the same problem. It'll make that copy not okay. So the insertions, they actually call them indels for insertion deletion. So an indel is like, we don't care if it's an insertion or deletion, it just messed it up in a way that is breaking. So yeah, insertions, small insertions can be just as big a problem. Now we have some variants in our group that maybe duplicate 3 nucleotides, and that just adds in an amino acid right in frame. So there's no protein truncating variant. It just, like, instead of 1,343, it has 1,344 amino acids in it. And where it should have gone like ACD, it goes ACCD, right? Like, and so that all it did was like shift one down, right? It added in one and shifted one down. That is essentially, that's not really called a missense, but in my mind, I kind of put that in the missense bucket because it's a full-length protein. It has one very small localized change that we're not quite sure what happens. So if it's a really small in-frame insertion or deletion, in my mind, I think of it more like a missense, even though that's not the right name for it.
Yeah, I think there were a lot of presentations yesterday about nonsense, or they call them PTVs sometimes as well, and missense variants. And basically, when there's a mutation, like, to drastically oversimplify, like, 2 things can happen. Either the protein just doesn't get made at all because it gets stopped, or a messed up version of the protein gets made. So if it doesn't get made at all, it's a nonsense. If it's a messed up version, it's a missense. And we know pretty well what happens if it's a nonsense. It just doesn't get made. There's not enough of it. And a huge section of the talks yesterday was what's happening with the missense. Like, what's the effect of having a messed up SYNGAP protein? That's what a lot of those presentations yesterday were about. The guy that was showing the structure of the proteins. That's something that we really need to figure it out, and it's very important for treatments because a lot of these treatments rely on increasing the amount of the SYNGAP protein that the kids have, and if that other protein is messed up in certain ways and we don't understand how, increasing that amount could lead to problems.
I think I've done pretty well not saying anything so far, and you guys are killing it. But this question is a huge one, and the way I've learned, I've given like hour-long lectures on this and then walked away and been like, damn it, I should have said this. Every mutation, broadly speaking, is either protein truncating, which would be nonsense, frame shift, indels, intronic, blah, blah, blah. They're all protein truncating. And they are assumed to behave the same way. Because they're protein truncating, the copy isn't complete, you have 50%. Good copy, bad copy. And we assume although I think over time we might evolve our position here. We assume that all protein truncating variants behave the same. Good copy, bad copy. Missense are exactly the opposite. Every single missense needs to be interrogated differently, and we assume will have its own unique action until we can figure out otherwise. Now, if you want to get nuanced, we could debate that, but the reason anyone in this room would care is every therapy, almost broadly speaking, every therapy that's under development right now will help the protein truncating mutants, but the missense mutants will be excluded from those therapies until we figure out what that missense mutation is doing. So I think, like, that's my soundbite where I think it boils down to. Don't go down the rabbit hole of, the letter, the amino acids, the whatever. Do I have a protein truncating mutation or do I have a missense mutation?
Yeah, and step one is, as a parent, understand the mutation that your child has and what it means and which one of those two buckets it falls into. That's important for you to understand what their treatment path is. And Mike's got a really good podcast episode about this. If you go through the history, he talks in detail about this and what you can do to prepare.
Kind of building off, Mike, what you just said, so I understand the concern around overexpression of SYNGAP. Are we aware of any kind of disorder that's observed where somebody has too much SYNGAP and we know what that looks like?
I think no. This is a frustratingly, we're frustrated that we don't know the answer to this question. It's been asked many times. Some people, we were looking at a paper yesterday where someone wrote, overexpressing SYNGAP is dangerous, but didn't cite any actual paper that indicates that. So I don't think we know the answer to this question. We really want to know it.
Aaron?
I mean, we do have, we know of one individual that has 3 copies of SYNGAP. So we do have some idea.
Yeah, I'd say we know they have 3 copies. We don't know what their expression level is.
Correct. Right.
So we know they have 3 copies and that they had clinical features close enough to us to be diagnosed with a pathogenic variant. So, but that doesn't mean that we know what the expression levels are in them. So we do have some anecdotal evidence from someone studying something else. They said they were studying something else and they're like, oh, I thought maybe I'd just increase SYNGAP in order to do this other thing that was, because they weren't thinking about us at all. They had their own, I think it's Alzheimer's research maybe. Yeah, it's tau, but isn't it Alzheimer's research? Yeah, so they were looking at tau protein in the brain and they're like, oh, maybe we can rescue this with increasing SYNGAP. And then they're like, wow, we really couldn't get the expression level up. We were trying in these cells and we just couldn't get it up. And so they're like, we think that that tau's really depressing, like down regulating the expression of it. So it's not clear to me how it works, but it's not as easy as like just put some more in and it'll be up. Because our body's super complicated.
Yeah, we need to figure this out. Janos Zamplini, who presented yesterday, I think it was the second presenter, he actually, with the nasal spray, managed to significantly overexpress SYNGAP in those mice, and he didn't see any.
I wouldn't say he overexpressed it. I'd say he delivered it. He delivered a lot of SYNGAP there, which is different than overexpressing it. Here you go.
Yes. Sorry. And the mice were okay, but yeah, we don't have the exact, we don't know.
I've done it in the frogs. Oh, yeah. So I've done it in the frogs where we can deliver a bucket load of SYNGAP. And I think it's going to be a matter of dose. I mean, you go way too high, there are plenty of defects when we do that in the frogs. So it's going to be like most of these genes, a Goldilocks problem. But.
What kind of fold is this? Like one? Like
more than that, 5-fold.
Five-fold. Okay.
Yeah.
That's a lot. Yeah.
Hi, good morning, everyone. My name is Paul Wong. I'm from the Simons Foundation, and if it's appropriate, I want to try to go back to the question about rescue and what it means. I don't know I don't know SYNGAP1 very well. I'm a developmental behavioral pediatrician. I used to see a lot of patients, but back when I was practicing medicine, people weren't getting diagnosed with SYNGAP1 or very many genetic things. Since then, I've worked in industry, working on studies for autism and Fragile X, and now I'm at the Simons Foundation. You'll hear about Simons Searchlight. Many of you probably know it already. We've also given a lot of grants to researchers who study SYNGAP1. Dr. Wilsey, Dr. Rumbaugh, you mentioned somebody else also this morning already, Dr. Dahlman. All of them have had grants from us to study SYNGAP1. As far as rescue goes, in mouse, it's incredible. So there are impairments, deficits that the knockout mouse or in SYNGAP1 and other conditions as well, that when researchers say they rescue it, they mean they completely bring it back to normal. But that's mouse. And the short answer to what that means for people, if those treatments ever get to people, is we don't know exactly how much of a difference it's going to make. But this is what we do know. We know that FDA is not going to approve something unless it is a clinically meaningful difference that it makes. Those are the words that they use. It has to really make a difference. And if I can give a couple of illustrations, one from an approved gene therapy, and then stories from research that's going on in other developmental genetic conditions, Angelman syndrome, Rett syndrome, actually a little bit about Fragile X as well. So there's a genetic form of blindness in which children lose the ability to see. And you can deliver the gene into the cells in the retina. It's approved by FDA. These kids basically really couldn't see. They could see light and dark, as I understand it. And what they did, and this goes to the point that JR was making earlier about endpoints in designing a trial, they had to design a special way to measure the improvement. These kids did not suddenly have 20/20 vision. It was not perfect, full rescue. But they designed literally an obstacle course. And they measured how long it took the kids to get through the obstacle course in dim light. So they did better. It really made a difference in their lives. They could navigate. They could go find things. They weren't bumping over things and tripping over things. It's not complete, full rescue, but it really made a difference in their lives. There's a drug that just got approved for Rett syndrome this year. It's not a gene therapy. It's a so-called small molecule. It's a pill. And as some of you might know, one of the characteristic problems in Rett syndrome is that the girls, it's almost all girls, they lose purposeful use of their hands, and they just put their hands together in front of their bodies and just they wring their hands for hours of a day. What I have heard from the neurologists who ran this study is that some of these girls, they start using their hands purposefully. So they'll start reaching for things, reaching for food that they want, reaching for a toy that they want, something like that. So it's obviously not full rescue, but it's meaningful. It's making a meaningful difference. So meanwhile, there are now studies going on for gene therapy, gene replacement, in conditions like Angelman's and Rett syndrome. And the stories from those studies, I think, also help us understand what rescue might entail. It's just stories, because this is not published yet. It's not even really been presented in a fully vetted scientific conference kind of way. But the story in Angelman's are that kids go from either having no words or just one or two words to saying short phrases. Or a great story, this is from Dr. Liz Berry-Kravis, who's at Rush University in Chicago. There's a girl with Angelman who loved playing in the water. If she could do that all day, every day, she'd be really, really happy. But if she didn't have someone standing right next to her, she would literally drown. She could not keep her head above water as much as she loved it. That girl is now swimming one lap. So she is not like her typically developing siblings, but the difference that the treatment seems to have made really seems to be meaningful to her and to her family. I think I'll just leave it at that. So, the bottom line is we don't know what kind of differences it'll make. We can hope that it'll really, really make a meaningful change.
I'll make a question. From each of your viewpoints, what is your biggest takeaway of the entire day yesterday?
Yeah, I mean, I said it already. I think the progress that's been made on the clinical side, for sure. But also, I looked at the list of attendees yesterday, and there were a lot of people from pharma in the room. They kind of keep a low profile, but they were there. There is intense interest in this disease, you know, from the industry, and these people don't come to these conferences unless they're very serious. So I think we're doing really, really well, and yeah, we just need to figure out how to make it go faster and how to make it easier for these companies to get these drugs to the market.
Yeah, I totally agree with Pavel, but I'll say a second thing, which is that I'm really impressed with how many people who are studying SYNGAP are talking to other people who are studying SYNGAP. And so there's a huge amount of collaboration, of crosstalk, of idea sharing, of, you know, just getting to ideas that are maybe new, and oh, maybe we could do this, and maybe we tried this thing with this other neuro that's a little similar, maybe we'll try that. And oh, we tried this thing with this other, maybe we'll try that. And so I'm really, really pleased with the sort of weight of all the brains that are thinking about SYNGAP. It's really, really impressive. And the amount of airtime that it gets between the people that are thinking about it, in their conversations. So it's so much more than it was when we were first diagnosed, and I knew about a couple papers and a couple people studying it at the time. We're just as, then it was like a couple I shouldn't say snowflakes. Then it was like a snowball, going down the mountain, and I feel like we're at like almost at avalanche, almost at avalanche velocity and volume right now.
I have a quick question. One of the big topics of some of the things our kids struggle with is sleep. And I know that's something that's measurable, as far as quality of sleep can be measured. And I'm curious if that's something that anyone's working on or you know of, and if it's,
Yeah.
Do you have anything?
Yeah. So actually, the person from Edinburgh yesterday, who was presenting about the EEGs also had a section on sleep, and he was citing some work that Lindsay Meisen had done in Edinburgh. She's studying sleep. There's a device. What's that? What's that device called? The sleep measurement device. I don't remember what it's called. So there's some device which they actually send to the kids, and they wear it at night, and yeah, good luck. That's the life of a SYNGAP parent. Yeah. You learn to recognize your kid's scream from a distance. So there is active work going on. Connie Smith Hicks has published something on sleep as well. So sleep is a measurable thing that you could reference in a clinical trial, like the improvement in sleep. So it's really, really important along with EEGs. So there is definitely work going on.
Yeah, I don't really have anything to add. I'm not sure what work is going on understanding sleep. I know there's a lot of work going on in understanding how much our kids sleep and whether they're in REM or not. Like my son has had a lot of sleep studies, and he really almost doesn't go into REM sleep. And so that as an endpoint, it's really interesting. I'm not sure people are definitely trying to affect seizures, right? Like there's a lot of anti-epilepsy medicines. I'm not sure that people are trying to affect sleep by itself, but I think that most of us sort of at a gut level believe that improvements will include improved sleep. Or maybe we're just desperate to hope. I don't know. but it seems reasonable, and it's definitely a measure that will be a meaningful, like back to what our friend Paul Yang, said about meaningful things to families. I mean, that's, depending on the age of your child, that could be number one, or maybe it's down at number three, but it's pretty high usually.
And he presented yesterday that the REM sleep in the mice is bad as well. And it's so great to have that measurable in the mice, because then you can fix it in the mice. You can give the mice the drugs. So yeah, it's a really important thing.
Okay, so for the families in the room, how many of you know what your genetic variant is? Sweet. Well done. How many have a tattoo of that? Yeah, nice. Nice. Two tattoos, all right. Of the variant? Yeah.
Peter's got one.
And Peter's got one, we know 3.
She's got one more.
The other thing that came to my mind was it's Fraser's study, who's watching the videos of the facial expressions, and I was curious if they could do a facial recognition software type thing, because we talked about some of the similarities in the way that our children look, and if there was some. I mean, facial recognition software is so advanced. I know on my iPhone it can pick out every picture of my dogs and her. So if there was something that we could identify in our kids that was significant enough that clinicians could help identify a Syngapian.
Yeah, it's such a good question, because you see some of the other kids and they do look so like, it's just striking that they look so similar. You know, my daughter, and we like, she looks very similar to a lot of the other Syngap kids. So I think anecdotally we like feel that as parents. And this topic of dysmorphia came up like yesterday a little bit. And sometimes it's listed as a symptom and sometimes it isn't. But I do believe that if we like applied some like large scale facial recognition technology to a bunch of like epilepsy kids, we might find something. I just don't know how feasible that is. Go ahead, Aaron. Oh.
Yeah, so there's an application called Face to Gene, and the goal of the Face to Gene that was developed by physicians that look at dysmorphology, and so there has been a lot of debate. So I've actually, Syngap early on did have some input into the Face to Gene application, so there is a database. I've actually have access to it, and I've used Jackson's, and Syngap doesn't come up first. Angelman will be up there, and some others that are very similar to Syngap. So it does exist, because the idea is in a lot of the world, they don't have the technologies, and the goal was to use Face to Gene to be able to identify rare diseases or through this application. Dr. Scheffer's paper does talk about some dysmorphic features in her paper, but when I've talked to a physician who specializes in dysmorphology, and she says, in Syngap there really isn't. So I think it's open for debate as we continue to have our patient population grow. But then how would we, again, the physicians really want to use that to make a diagnosis, even maybe over genetics, because it's easy. But then we have, so there is the technology that's there. We've had partnerships with them in the past. We haven't done a lot. I don't know what the application's doing today, but there could be opportunities to expand that database, because anytime you can expand a database, like the work we're doing with KKI, to expand, and I think that probably goes into that study that KKI is doing, is to expand the genetic profiles of diseases, and maybe part of that is facial feature recognition that would be part of that database. So,
Yeah, and like just finding more patients is critical as we know. And one of the things that was really exciting yesterday was the machine learning model that was applied to the EEGs. You know, is the EEG signature is extremely detailed, and so if there is something that's SYNGAP specific or SYNGAP like there, and there's so much EEG data, if we could figure out a way to apply that model to a large range of EEGs, I think that would be a really powerful systematic way of finding more patients, not just for SYNGAP, by the way, for probably for a lot of diseases. I see Jim there from STXBP1. So that, you know, we need to like try all of these types of techniques to try to find more patients in the community. There's so many undiagnosed patients out there.
Anybody else?
You're welcome. Can you hear me? So I thought yesterday was really great. What kind of struck me was how little there was discussed ultimately about behaviors. They had that one presentation that talked about caregivers care about this, and clinicians, right? And so I'd be interested in your guys' take. There was the discussion. I can't remember the name of the guy. He was talking about neurobehavioral connections. About what do we know right now about why our kids have the behaviors, so that then we can follow that down. I mean, they talked about some different surveys, and because you can control sleep with clonidine or different medications. You can control, to a certain extent, seizures, and a lot of people are working on that. But behaviors, what, right? And I'd be curious on your take about that connection, what we've learned, and where it's going.
So I think for behaviors, you mean from the scientific perspective or from a personal experience perspective?
I'm talking about it from a scientific perspective and as relating to the biological basis for behavior, right? There's something going on in our kids' brains. It's not just poor parenting, as I like to tell people. I am not a horrible parent, right? And everyone understands that, right? Yeah. You almost wish you had a little card that says when your child is freaking out, you can just hand them something and be like, just don't call the cops on me, right? As I try to lift my child and put him in the car. So I'm curious about, what do you think in terms of the neurological, the biological basis for behavior, the progress that's been made on that, so that we can ultimately close that gap, right, between caregivers' perspectives and scientists' perspectives?
I have a lot to say on it, but I want to know, like, Jim, do you have anything to say on it, or Mike? Does somebody who's going to talk shorter and with data, shorter with data? Yeah, Mike has something to say. No, Mike has it. Well, Mike has his hand up, so Jim, go.
I wish I could actually answer that question. That's an excellent question, but ultimately difficult to assess, right? So there are animal models out there, but I don't think anybody's really assessing that particular type of behavior. And I can't recall if anybody's done any, so along those lines of trying to look in mice, so rearing and parenting in mice. So I don't know, are the female SYNGAP mice able to properly nurture their pups, right? So these are the kind of some of the behavioral ways in looking at the animal models, at least. And I don't know if anybody's really delving into those kind of studies. I mean, there are some other sort of behavioral studies that you can do as far as looking at fear and a couple other sorts of higher level learning that get to that. But for those exact type of behavioral issues I think you're talking about, animals are limited as to what you can do with that.
have an equally unsatisfying answer, and then I'm going to plant a question that I was hoping somebody else would ask. But so Jim's right. This is understudied. I mean, we funded a mouse, and then we funded that mouse being characterized. And then as we were talking about characterizing this mouse, behavior came up. And we were talking to Ben Prosser and Jim, we were on these emails, we're talking to some very senior people, talking to Jackson Labs, who ostensibly is the biggest mouse expert in America. And we were like, do a behavioral assay. You said this, JR. And they're like, what would that look like? So I emailed Rick Huganier, who made the first Syngap mouse. And I was like, hey, what kind of behavioral? He said, I don't know. And he emailed Ingo, who was here yesterday. This whole email chain came back to me, and I was blown away that nobody knew the answer to this question, right? And the thing I've been saying for years, and this is why I love those beautiful tables that Chop did yesterday, showing the behavior and the autistic features on Syngap and how different we are. And finally, clinicians are getting it. We are facing some of the hardest behaviors of any neurodevelopmental disease consistently, right? Dravet kids, other kids, STX, some STX kids maybe, they have challenging behaviors. But I think every Syngap parent, we all know it, and to the parents of young kids in the room, I'm sorry you're hearing me say this, but Gasper, we were talking about this last night. As your kids get older, they will get harder. They will break things. And the sooner we tell you that, the better, because it'll change the furniture you buy. That really pretty thing made of glass, don't buy it. It's going down. But so I think your question is right, Heather, and I think we need to consistently ask this question, and we should have, hindsight's 2020, we probably should have asked it more yesterday. What about behaviors? Are you studying behaviors? Like this program at CHOP is huge, because Ingo Helbig, I've been on bended knee for years. Ingo studies Syngap. Oh yes, Mike. Does STX, BP1 and Syngap. Finally, someone gives him $12.5 million. Now he's seeing 38 kids. Now he's like, you know what, Mike, I share your me for a year. You're right. These behaviors are tough. I'm like, yeah. So here's the question I want to ask you guys to talk about, because I want to hear someone else answer this. We heard can you talk about repurposed drugs, ASOs, because we heard from 2 ASO companies yesterday, right? Praxis and Stoke. And these other cool gene therapy things, which I think are delivered through some AAV thing. And could you please help every parent in this room frame small molecules, which is repurposed, ASOs, and AAVs in the context of when it could actually happen, and then maybe touch on one and done.
Yeah, sure. So, so I think we should start by saying there's gene therapies and there's other disease modifying drugs, and the thing you're trying to fix can be different. You could be trying to make more Syngap, or you could be trying to you could be trying to resolve some of the problems that happen because there's not enough Syngap, as we discussed earlier. So there's different types of drugs. But let's just say that we're talking about increasing the levels of Syngap. So there's obviously different ways you can do that. So a small molecule, which is what RareBase presented yesterday, would be hopefully an FDA-approved drug, like a pill, right? You take pills and you drink them, and they're there. That would increase your Syngap levels. So those are oftentimes already approved by FDA. Maybe they're already approved for pediatrics, so it's already safe for children to take them. And as a result, the path to getting those is very, very fast. I mean, Zach Grinspan yesterday said you can just prescribe a drug for off-label use. So that's really, that would really be great from a speed perspective. That's a drug that you could give to your kid almost immediately. But finding a drug that's approved for some other indication, like an antihistamine, that also increases Syngap levels would require like an enormous amount of luck. And we're trying to find those types of drugs. So that's the fastest path. ASOs are a type of gene therapy which will increase the amount of Syngap that you have using some of these genetic tricks that were presented yesterday. And ASOs are proven. There are approved ASOs out there in the market for other diseases. So it's not a pipe dream. It's a real thing that's approved. And they've proven to be effective. And that's what Stoke and Praxis presented yesterday, are ASOs. So they're very, very close to being to being available. The Dravet one that Stoke is doing is in clinical trials now, and we're hopeful that Syngap will be there next. The problem with ASOs is they don't stay in the system forever. You have to go and get them put in every 4 months, every 6 months, typically through a spinal tap. So it's like not great. I'm sure many of us would make that choice, and if it made our kid better, we would take them every 4 months. But it's not ideal. And so the sort of next generation set of drugs are AAVs, which are one and done, one-shot drugs. So they basically actually modify the behavior of the cells so that they make more Syngap forever. And that's great because you only do the one shot and it's done, but it's also extremely risky because if you mess up, then it's permanent. Whereas with an ASO, if it's something causing some adverse effects, it'll just leave the system after a certain amount of time. So the approval path for AAVs and getting them to market is a lot more tricky, but it's coming, it's going to happen, and we're paying attention to it. But it's definitely sort of the next phase. So I think we're really focused on getting the small molecules and ASOs going as fast as we can because we feel those are the ones that are going to get to the kids as fast as possible. But we are paying attention to the AAVs as well.
Yeah. Do you guys have any questions about what those kinds of drugs are, about the 3 different types? Suzanne?
I'm never going to wrap my brain around this properly. I apologize. The small molecules kind of falls in the drug category. And then it sounds like ASO and AAVs are drugs, but they fall in the gene therapy category?
Yeah, I'd say precision medicine.
Okay.
Yeah.
And
They're
then
precision because they will be specific to Syngap. So
Okay.
just because another gene has an ASO, that means the technology is there. Everything is there, but their ASO isn't going to fix Syngap. We have
Right.
to have our ASO, and it's going to recognize something in our gene.
Okay, and then maybe I shouldn't ask this, but then compared to all this, how does CRISPR and that kind of stuff compare to all this?
Yeah, so that would be maybe something that went into an AAV, although I think there's other things going into an AAV before CRISPR.
Okay.
Probably, yeah.
But that falls in this
Oh,
general.
let Jim say it, yeah.
Sorry.
So it can be very complicated, and so one way to think about it is that a repurposed drug and an ASO are still drugs as far as like to think about it. It's something that you would take on a repeated basis, right? So if you have high blood pressure, you take a high blood pressure pill every day, and you need to take that in order to have the effect. For an ASO, because as was mentioned, the ASOs don't exist. They disappear in the body. They're degenerated in the body. So those have to be reapplied on a regular basis, and it's going to depend upon the ASO and the gene and other issues, biology, as to how often you'll have to take those. But they will be the kind of the first precision medicine, as JR was saying, that will be available, I'm sure, for Syngap. They're the easiest. They're easy to manufacture. They're the way to get through regulatory approval, the FDA approval. That pathway is well known. It's been traveled. And so I would think that an ASO could be available for Syngap within the next couple years, probably, in clinical trial. And so the other is gene therapy, which was mentioned. So when we're talking about gene therapy, there are a couple different things that you can do. So you can replace or enhance the gene itself, which is mean you put in a working copy of the Syngap gene into the body, right? And so that takes over the function of the mutated gene. And in that case, the idea is you're overexpressing Syngap. So as long as overexpressing Syngap isn't bad, you're fine. And I don't know if there's any evidence that suggests that we already had this conversation, but I don't think there's any evidence that suggests that overexpressing SYNGAP is bad. And so that's one type of gene therapy. Another type of gene therapy is to add in what we call, for lack of a better description, molecular machinery, in order to actually go in and fix the mutation in SYNGAP. So whatever mutation you have, your variant that your child has in SYNGAP, there are different ways of fixing that. So you're repairing the gene. And the idea behind repairing the gene is once you repair the gene, then it will function normally, and there won't be any chance of overexpression or anything else like that, because it'll function in the same way that the normal other allele functions. And so those types of gene therapy, both gene replacement therapy and gene editing, which is the second thing I just mentioned there. So both of those are further behind ASO development, but both of those are realistic within probably the next 5 to 10 years to see. So the first gene editing with CRISPR, and that's what CRISPR does, it's a gene editing approach, was just approved by the FDA. And so gene replacement therapies have already been approved by the FDA. So all of these technologies are available. All of them have been approved for different disorders. They are coming along on the pipeline. And chances are, ultimately there will be a number of different therapy options in 10 or 15 years. And, like any other drug, some may work better for some of the SYNGAP population, others may work better for other set of populations.
And so then the editing and replacement is considered to be AAV? Sorry.
So yeah. So AAV, which is adeno-associated virus, is a delivery mechanism. So this is a different question. And so whenever you're delivering a gene, like a replacement gene, so if you're delivering a working copy of the SYNGAP gene to the body, or you're delivering a gene editing molecule like CRISPR to the body, you need some way of delivering it to the body. And there are a number of different ways that you can deliver these to the body. You could just inject the gene directly into cells, but that's difficult to do. And so what has been used traditionally are viruses, because viruses evolved basically to infect cells and to express their own gene in those cells. And so AAV is a virus, and basically what people do is they take the virus, in this case AAV, but there have been numerous different viruses that have been used to deliver genes throughout gene therapy research. But the idea is you take the genes out of the normal viral genes out of the virus, and you put in the gene that you want to put in. So if it's SYNGAP, you put in that gene, or you put in the DNA to make CRISPR into the virus. Then you inject the virus infects cells, and then it makes the gene. It causes the cell to make the gene, just like the normal virus causes a cell to make viral genes.
I just want to sort of simplify this, because the trouble with either asking JR or Jim or anyone who begins with J, apparently, a question is they get too complicated. There are 3 buckets for mortals to think about. Small molecule, as opposed to large molecule. A large molecule is a biologic. It's the kind of drugs you hear about for cancer. Large molecules are not in play. But the reason we say small molecule is every drug you've probably ever taken in your entire life has been a small molecule. It is a small drug. You stick it in your mouth. It goes into your stomach. It goes into your blood. Your liver cleans a ton of it out. Some of it gets to where it's going and fixes the problem, right? This is what Dr. Chow is doing, and I shouldn't be talking, he should, right? Small molecules are what we are hoping to find and help our kids ameliorate symptoms as fast as possible because we can put them in tomorrow. And when I said repeated here, I'm sure there's a better word for it. As was mentioned, you take a drug, your body cleans it, your body excretes it, you got to take the drug again the next day, right? Repeated dose through the mouth, the liver takes a hit. ASOs, why is it a spinal tap? It's a spinal tap because if you stick it in the mouth, you could drink an ASO, and all you're going to do is screw up your liver because your liver is going to absorb 90% of it, and you have to make a ton of it. And you want it to get to the brain, right? So 100 units go in the mouth, however many units hit the liver, 10 units make it to the blood, 1 unit makes it to the brain. 100 to 1, not good. I'm making those numbers up. So with an ASO, the way this is, I'm making up words, which is probably killing the PhDs in the room, but I'm trying to talk to mortals here. This is hitting the whole system, right? As JR said, this is precise. We all know SYNGAP in our kid's head is busted. So we want to get to the brain precisely. And we don't want to mess with the liver, which is why it's spinal tap, right? Because we want to bypass the liver. How do you bypass the liver? You go into the cerebrospinal fluid and you get it up to the brain directly. But you have to do that in a repeated fashion every 3 to 4 months because the drug is consumed, right? And then Jim mentioned virus. Next level, the virus gets in and the virus spreads. Good news, the virus gets in and the virus spreads. Bad news, the body knows how to deal with viruses. It forms an immune response. So the next time you put in that virus, the body will take it out. One and done. Which means titration is a problem. If you put in too much, what goes wrong? We don't know yet. We haven't done it to a human. If you put in too little, what goes wrong? Well, maybe nothing, but maybe not enough goes right, and you've just used that virus. Because when you start talking about AAVs, you're talking about delivery and cargo, right? There are multiple companies thinking about using AAV9 to deliver what? Something that does something that makes SYNGAP go up. So when we start talking about AAVs and CRISPR and all this madness, and like yesterday we talked about exosomes with Zemplenty, right? Exosomes would be another delivery mechanism. What would we deliver? Something else. But, we can go way deep and dazzle you with profound details. But the punchline for us as parents raising kids who are getting older is if we can find a small molecule, we can use it tomorrow. We are familiar with this model. ASOs are new. We've used them for spinal muscular atrophy. And we're seeing them go into kids for Dravet, and we heard from Praxis yesterday, who I think is sticking them in kids for, SCN2A. But we're at the very beginning of this type of medicine for the brain. And the reason I think it's worth every parent in here getting this through your heads, getting this through our collective heads. Right now, it's like, this is weird science stuff that we talked about before coffee. In 1 or 2, maybe 3 years, companies will be calling you. I will be calling you saying, you have a child who is of age that they could participate in a trial. Would you like to fly to a hospital and stick a needle in your child's spine to take a medicine that no one's put in a human before? We're going to have that conversation. So you've got to get your heads wrapped around what this is, and you've got to pay attention to where it's happening, because this is coming. And then in a few years after that, you heard from Tevard yesterday, very exciting. That'll be delivered by a viral vector. And here's the thing about viral vector. Tevard has got their tRNA suppressor in an AAV9. Probably correct? Okay. There are other companies, a lot of other companies, who also have things in AAV9. So not only do you have to get your head wrapped around, am I comfortable putting an AAV9 in my kid's brain? You've got to be comfortable with, if I only get to put an AAV9 in my kid's brain once, do I want to do it with Tevard's technology? Or do I want to do it with Company A's technology or Company B's technology? And those are just the 2 NDAs I can think of that I'm under right now, who are also using AAV9. Right? So this is not a theoretical conversation. We are not here because anyone wants to be out of bed right now. These are therapeutic approaches you can and hopefully will have a chance to do to your loved one. So I think when you start CRISPR and prime editing, we had a huge discussion at the board, a very lively debate, and then I'll shut up. let's spend a bunch of money on prime editing. And I said, no way. A, it's a lot of money. B, there's all this other stuff that's going to happen first. And by the way, do you remember the biomarker conversation? Nobody knows what to measure when we do any of these right now. That's why I was so excited when I introduced the EEG guy. When we do this or this or this, what will we measure? Are we going to have Sydney say, last week Emmett had 20 seizures and then we gave him the drug and this week Emmett had 5 seizures? Is Sydney's word good enough for the FDA? It's not. Sorry. The FDA wants something they can measure, something that's validated, something that's been published. That's why Fraser was important, right? So I'm sorry to send us down this rabbit hole, but this is real, guys. Like, we throw a prime editing, CRISPR, blah, blah, blah. Which drug will you put in your kid? This is coming in a very reasonable number of months. So sink your teeth into this and ask a lot of questions. And if you meet industry people, say to them, are you doing an ASO or an AAV? How many years? Be polite, thank them for being here, but push them just a little bit. Because this is not a theoretical anymore. 5 years ago, it was a one day maybe. This is coming, and it's coming fast.